Okay, our next presenter is Sara Nedrich from the Dow Chemical Company. She's another environmental toxicologist.
All right, hello. I'm just trying to get my screens figured out here.
We're still figuring stuff out on our end too, so you're good. Oh, we can see the slides.
Ashley, that was great, by the way. If you ever want another field site, please come to Ann Arbor and get some data on the Huron. All right. So let's see. I think I almost got it here. All right. So yeah, my name is Sara Nedrich. I'll be presenting today on 3 case studies where chemical mixtures were an important driver for protecting ecological health. I'm going to go through how the work was done, for what reasons, and how we can continue to encourage these types of assessments in the future. So I'll start with a high-level introduction to mixtures assessment, though it sounds like you're all pros, and highlight some tools currently being used to conduct assessments. I have 3 case studies on metals, herbicides, and neonicotinoids. These are going to be high level, and then I've listed some technologies here we'll go over. And then I'm going to wrap up with some key takeaways. The last time I presented at this conference, I was working for Eagle, presenting on 6-PPD quinone. I've since moved on to Dow Chemical. I'm working as an aquatic specialist, environmental toxicologist, and risk assessor in support of their global environmental remediation function. The collaborators on this talk are many, since I'm covering 3 projects. In order, Jacobs and Olson completed the metals Pit Lake study. University of Saskatchewan and Geosyntec are our current partners on the herbicide mixture project, and the neonicotinoid study was completed while I was with Eagle. So thank you to our partners. All right, so when it comes down to it, it's a fact that we're living in a chemical soup. The problem is, how do we determine whether it's chicken noodle or toxic stew? And yes, I'm going to go further with this stew analogy. So how do we determine if a stew is toxic? Standard lab-based toxicity tests evaluate each chemical individually and assume after releasing it to the environment, it will behave the same or have the same toxicity. Generic criteria promulgated under the Clean Water Act are by default based off this assumption. This is kind of like eating the ingredients of a stew and assuming it will taste like stew. Both of these pictures are of beef stew, but would you ever eat the flour on its own followed by the beef broth? Much like with cooking, it matters the way chemicals interact, like flavors blending in a crock pot. It matters what conditions they're exposed to, like light and heat. And in the work I present today, we're not assessing the parts, but the whole stew. At a high level, there's 2 broad categories for determining if stew is toxic. There's sort of the global generic frameworks, which are established from standardized tox lab studies. And there are more local and site-specific frameworks, which include risk assessment completed to assess local effects. They each have their pros and cons listed here, but no time to go into, with generic evaluations providing the most value in predicting or preventing effects of new chemicals, whereas site-specific criteria are best implemented when the pollution has already occurred and effects need assessed. I don't have time to go over all the different methods applied in detail for mixture analysis, but I've listed them out on this slide for reference. And I'll point you toward EPA's recently published framework. See if I can get, which is right here. I'll cite that in references so you can look that up. Today, we're mostly focusing on these local frameworks. Oh, and I'm supposed to click. Come on. Boxed in green. This is why I don't put animations, because I forget they're there. All right, so on to case studies. So if you guys get, drift off and get a little sleepy, just know the icons will tell you which one we're on. There she is. So on our first case study, we're going to look at a pit lake at a former mining site, pictured here. Pit lakes are formed when open pit mining operations end and that excavation fills with ground and surface water. The water in this lake is prone to low pH, so it's very acidic, and has high metals, sulfate, and total dissolved solids. For this specific lake, we conducted whole effluent toxicity tests, also known as wet tests, on a monthly basis for a year. Wet tests are lab-based studies that take site water at the outflow of the lake and expose an aquatic organism for measured adverse effect, like effects to survival, growth, or reproduction of the organism. Wet organisms in the study included fathead minnow and Ceratophrenia dubia and looked at acute and chronic endpoints. Based on the wet results and metal concentrations, we were unable to consistently predict an effect from any single metal concentration. So additional modeling work was done. Okay. Reproduction of Ceratophrenia dubia is where we saw our most sensitive adverse effect, and that's shown in this graph here. You can see somewhat of a relationship here with zinc, where zinc is above 100 micrograms per liter. You can also see that those dates correlating with low reproduction. Comparing the toxic units generated between a few of the metals, you can see zinc was predicted to be the most hazardous metal here in these purple triangles, although manganese was also elevated in the green circles here in our December sample. Toxic units are generally derived by dividing a detected concentration by an adverse effect level. So in this case, the toxic units were derived by the MBAT model, which is a metals bioavailability assessment tool. It is very similar to EPA's biotic ligand model, for those of you familiar with that. Metals are dependent on pH, water hardness, dissolved organic carbon, and a few other chemical parameters to predict their toxicity, which is why using a model such as MBAT or BLM is important for predicting toxicity. In this slide, I tried to overlay two PCA diagrams over each other, so they're both a little bit tricky to read. But you can see in this, the reproduction is decreased as these red dots where the metals concentrations are greatest. And it's most strongly correlated with the sum toxic units, as shown in this figure here, where we get an R squared of 0.94. We did find that zinc was a strong predictor of effect, but that some toxic unit was much stronger. Of all the different chemical mixtures possible that you find in the environment, the best developed models are for metals. These metal models provide a roadmap for how we can build other models for specific types of toxicants and include the bits of chemical soup that are relevant for predicting mixture toxicity. Okay, for our next case study, we have a site that's been a disposal area for a diverse mixture of agrochemicals, including phenoxyacid herbicides, chlorophenols, and VOCs. We are designing a biostimulation study to assess microbial efficacy to degrade our chlorinated compounds at the site, hoping that it might decrease our concentrations of our target species. We're concerned, however, that if those parent compound concentrations decrease, we may still be producing daughter or breakdown products that could make the situation potentially worse or more toxic. To add to that, a recent study on human cell line metabolism, led by Mary Ella Hashim at University of Saskatchewan, showed pesticide mixture synergism or enhanced toxicity for mixtures. So the objective of our study is to see if biostimulation will decrease soil toxicity. The figure shows at a high level the biostim conceptual model. So to biostimulate a soil, you essentially add nutrients and a carbon source to encourage microbial degradation. So compare that to natural attenuation, which is the natural biological consumption of products in the soil, compared to a sterile or non-biological control. On this slide, I focus on the toxicity testing plans for this study. Note that the project has just kicked off, so I won't have results for a year or two. We'll be doing toxicity testing of soil and water before and after the bench scale biostimulation treatment is conducted. Our bioanalytical toolbox will include in vitro cell assays to target fish embryo and target organ toxicity. And you can see the number of cell lines we've selected here to include in the study. We'll also be doing an in vivo fish embryo assay with rainbow trout. That will include early life cycle, acute, and subchronic exposures. And the coolest part of this all is we're also doing a focused transcriptome analysis using qPCR arrays, which is the EcoToxChip method. I did want to talk a little bit more about EcoToxChips, since this is a newer technology. From doing in vivo and cell-based assays on rainbow trout, we compare adverse effects up here with genetic fingerprinting. Using the existing EcoTox modules to read specific genomic clusters that are damaged during the tox testing, we can then predict impacts to biological processes, and then confirm the impact of those damaged processes on apical outcomes, such as survival, growth, and reproduction. Once fully developed, EcoToxChip could be used for biomonitoring of mixtures without relying on live animal testing, so sort of like a next stage wet test. For my last case study, I'm focusing on watershed scale assessments of neonicotinoids. Neonics are nicotine-based pesticides used in broadleaf applications and seed coatings for corn and soybean fields throughout Michigan, which is where this study was conducted, as you can see by this pretty little map here. Regulatory criteria did not exist for neonicotinoids at the time of the study. However, we had lots of articles showing chronic effects to organisms at low levels, and that's part per trillion levels. So our question was, is there potential for aquatic organisms to be adversely affected by neonics in the Saginaw watershed? We conducted surrogate biomonitoring using polar organic integrative samplers, also known as POCIS, which are pictured here to the right. When I say biomonitoring, POCIS have been found for some chemicals to have direct comparisons for uptake into fish tissue. Cervani et al. 2016 found this for PFAS in particular. We also collected grab surface samples monthly from the watershed we were testing. We also developed aquatic life values with standard lab tox test data pursuant to the state regs or Rule 57. In some of the Saginaw watershed, the developed imidacloprid FCVs, particularly for imidacloprid of 29, was exceeded, as you can see in this table here. To complicate this matter, while our field study was going on, an article by Travis Schmidt in Science Ecology was published showing a synergistic effect between clethianidin and imidacloprid at high nominal concentrations, around 500 nanograms per liter. However, an additive response was observed at lower doses of 50 nanograms per liter. Because one study is insufficient to change an FCV or to develop one, we weren't able to include that in our water quality guidance. However, in retrospect, it would have been really awesome, hindsight 2020, if our study had included biological monitoring, because then that could have been used to help validate the FCV that we finally came up with. But we lacked the time and funding to do so. Okay, so that was a lot. I hope that you are able to take away from this talk that chemical mixtures do matter. I've highlighted 3 examples where this was the case. It's important to remember that mixture toxicity is local and holistic. And by that, I mean it goes beyond summing the parts, but includes the whole stew. Local evaluations have the tendency to uncover truth that global models can miss. And third, we have tools, but need lower cost options to address mixture toxicity in the environment. Lastly, we need regulatory flexibility to develop site-specific criteria. This will ensure that environmental remediation projects are not over or under conservative and are done in the most economical and environmentally protective way. I have no clue how I am on time, but these are some of the references that I cited. If you are one of these authors and in the audience, thank you so much for your work. And that's it. Thank you.
Great. Thank you.